According to the Canadian Cancer Society in 2013, every hour of every day, an average of 21 Canadians will be diagnosed with cancer. 9 of them will die from the disease. Now, that's a pretty scary fact if you ask me. One of the factors that can influence your chances of getting cancer is your genetics. However, even if nobody in your family has ever experienced cancer, every single one of us has genes called oncogenes. Oncogenes are specific genes in a cell that have the potential and ability to transform a healthy cell into a cancerous one. They do this because they have a mutation in their healthy form called a proto-oncogene. Proto-oncogenes are integral genes in a cell that encode for proteins involved in signal transduction pathways and cell development. So, as you can imagine, if one of those genes were to become mutated, the cell adopts cancer-like behavior and loses control of important processes. So, some of the common causes of oncogenes is point and deletion mutations, chromosomal rearrangement, and gene amplification. Come with me, guys, to learn more.
Now let's take a quick second to talk about the past.
The first oncogenes were actually discovered through retroviral studies. The first oncogene that was confirmed was known as SARC, and it was actually found in a chicken retrovirus. And it was actually that the SARC proto-oncogene was actually very similar to the SARC oncogene. It was later analyzed and found that the SARC proto-oncogene had actually been captured by a virus and had undergone mutation to become the SARC oncogene. It was also later discovered that retroviral oncogenes had actually been derived from normal, healthy cellular genes from the genome of the host. But that's enough of the past.
Let's go back to the future.
Cancer's mutations occur throughout the cell and target a variety of pathways which are involved in the cell cycle and cell growth regulation, such as the GPCR, cyclins, tyrosine kinase receptors, and other pathways too numerous to mention. Some of the most common oncogenes target RAS-H, RAS-K, and RAS-N, which are found in the tyrosine kinase receptor pathway. So let's take a look at how these oncogenes affect the cell.
The RAS protein plays a key role in the mitogenic activity of the cell. When the RAS protein contains normal proto-oncogenes, it cycles between active and inactive states, active when bound to GTP and inactive when bound to GDP. RAS is inactivated by a GTPase activating protein, or GAP, which hydrolyzes GTP to GDP. If the RAS proto-oncogene mutates to a RAS oncogene, it is less responsive to GAP, which means it is constantly active. This means the downstream effects are constantly turned on, such as constant activation of transcription factors, for example. This could lead to unregulated cell replication and growth, or in other words, cancer.
So in conclusion, oncogene research allows us to better understand the genetic basis for the development of various types of cancers.
Oncogene studies are being applied diagnostically to help determine the predisposition of an individual to disease. They also aid in the accurate staging of disease, developing markers to detect residual disease and therapies such as oncogene protein antibodies or gene therapy.
Again, an oncogene is a mutated form of a proto-oncogene normally involved in regulated cell growth. These oncogenes were first discovered in retroviruses and can form via mutations, chromosomal rearrangements, or mRNA stability.
Abnormally activated RAS proteins are an important oncogenic driver. The pathway involved can dysregulate the cell cycle, leading to uncontrolled cell growth and division. Pathways using GPCRs and cyclins are also involved.
So studying oncogenes is important to further research into the understanding of the molecular basis of cancer and one day to hopefully cure it.
Hello everyone, this is the credits to our fantastic video. I hope you enjoyed it very well. So we're just going to show you a bit of the equipment that we used to film this. So this is the boomstick, which is basically a selfie stick and it's broken, so we duct taped it. And this is from the library. So this is the recording device we use to record the audio. This is the microphone, which belongs to Manel. And this is our camera setup. So as you can see, we have the camera over here, we have the tripod, and then we used a chair from the university as our dolly because we couldn't find ours. So we have we duct taped the 2 front ends of the tripod onto the desk, and we have 2 backpacks along with a physiology textbook to hold the 3rd part of the tripod. And as you can see, if you look down, it moves very well, although it sometimes does that. And a special mention to my brother here, Tawheed. He's my twin, although fraternal, and he helped us with filming this video. So we thank him a lot, and we hope you enjoyed it. My mind is stuck in the camera. Bye.